Welcome and thanks for joining us for today's Oriental Institute Collection Series talk. I'm Kirsten Newman, curator at the OI Museum, and I'll be hosting today's talk. The Oriental Institute of the University of Chicago is both a pioneering interdisciplinary research center and a world-renowned museum whose aim is to understand, reveal, and protect the earliest civilizations of the ancient Middle East. With 350,000 artifacts excavated mainly by OI archaeologists, the OI Museum houses the largest collection of ancient Middle Eastern artifacts in the United States. Since its founding in 1919, a time when the Middle East was called the Orient, the OI has pioneered innovative excavations and comprehensive dictionary projects that serve as cultural encyclopedias of ancient civilizations. Of the 350,000 artifacts that make up the OI Museum collection, about 5,000 are on permanent display in the main galleries, which are divided up in regions of the ancient Middle East. In addition to the artifacts, the OI Museum collection also encompasses an expansive archive collection composed primarily of materials related to the OI expeditions of the early 20th century. For example, field journals, object logs, and archival photographs. Additionally, the OI collection also includes more contemporary pieces, including paintings of archaeological sites, as well as photographs. An example of the last is a group of 6 large-scale paintings by renowned American artist Joseph Lyndon Smith, painted at the site of Persepolis in 1935, and which are the focus of our current special exhibition, which we welcome you to visit, entitled Joseph Lyndon Smith, The Persepolis Paintings, on display through August. So without further ado, a few words on our speaker this evening. Hannah is a postdoctoral fellow at the University of Chicago, working jointly with geneticist Mansa Raghavan and John Novembre in the Department of Human Genetics, and archaeologists James Osborne and David Schlone in the Department of Near Eastern Languages and Civilizations. Her PhD research has focused on using ancient DNA to study increasing trans-Mediterranean mobility patterns in the Iron Age and Roman Mediterranean world. Other past projects include an archaeogenetic analysis of the dispersal of several domesticated crops, including taro and broomcorn millet, and a study of the human impacts of aridification of the Saharan desert over the last 8,000 years at Gobero. She advocates for ethical and critical approaches to ancient DNA research, and her course, Bioethics in Ancient DNA, will be offered in fall 2022 here at UChicago. So now I will pass it over to Hannah to speak on From Wheat to Watermelon, Clues from Ancient DNA about Food and Diet in the Ancient World.
Awesome. Thank you so much, Kirsten. It's such an honor to be here today giving a collections talk. And before I get started, I want to acknowledge that the land that the University of Chicago is on is the original homelands and traditional territory of many indigenous nations, including the Kickapoo, the Peoria, the Potawatomi, the Miami, the Ottawa, and the Ho-Chunk people. These lands were the home of these native nations prior to their forced removal and relocation, and these lands continued to be embedded with the rich histories and struggle for survivals of each nation. So today, I'm really excited to be here giving this talk, as I mentioned before, about how we can use ancient DNA to better understand the domestication of plants and animals. We'll be taking a look at how we have changed plants and animals through this process, and also how they have changed us. I have the great opportunity today to be able to show you artifacts from the collections here at the OI. As you know, at many museums, many of the objects that are housed there aren't actually on display, but are in collections, and this is a wonderful chance for you to interact with them and them to interact with you and such. And so what I'll be showing today are objects that represent food and food preparation in the ancient world. It'll sort of be a 2-section talk. So there'll be a point in the middle to show some objects, and then again at the end to see objects and questions. And this talk highlights research that is happening throughout the field. It's not just my work. I want to make sure you all know that. So as I go through, I'll mention the research and the authors on each of these projects so you can check out their papers and books if you're interested in learning more. And at the end, I'll talk a little bit about my own research, so I'll let you know when that is as well. All right, so before we get started, I just want to jump into a little bit of the logistics of how ancient DNA research is done. So you'll start with an archaeological material. In this case, it might be parts of a plant, such as a leaf or a seed or a bone from, say, cattle or sheep, and you'll turn it into a powder. There's a couple different ways to do that. For a leaf, you might use liquid nitrogen to make it brittle. And then something like a mortar and pestle, you might also use a drill on something like bone. And then you go through this extraction procedure. It takes about a week. And it's, I like to describe it as sort of mixing a whole bunch of clear liquids together and hoping you did it right. And then at the end of the week, you'll find out if you did or not. And once it goes through that, you have what are called libraries, which are essentially sequences of ancient DNA that you've appended adapters and indices to. And what these indices let you do is basically tell different organisms apart or different samples apart. So if you've, say, extracted libraries from two separate leaves, when you sequence them, you'll put them in a pool together, and then you'll get data back and you'll be able to demultiplex them using these barcodes that you've put on the end. And the barcodes are made out of synthetic DNA. So they sort of have the same bases, the A, G, T, and C that we think of for DNA, but they are synthetic, so they don't match anything that's in a living organism. And then you do sequencing using one of these machines that kind of looks like a refrigerator. It'll run for 24 hours and then you get your data. And once you get your data, and this is a picture of a clean room. This one is at the University of Dublin, but we also have one here on campus at the University of Chicago, Dr. Raghavan's Ancient DNA Lab, and I have the opportunity to work there. But you can see that the person in this photo, this is one of my colleagues, is dressed sort of head to toe in this Tyvek suit, and she has gloves on. She actually has several layers of gloves on and a face mask. And actually the reason for this is we want to protect what we're working on from our own DNA, from contamination. So a lot of times when people wear these, it's to protect themselves from chemicals. But in this case, it's actually to protect the specimens, the material that we're working with from us, because we're sort of the biggest potential source of contamination, especially if we're working on human DNA. So, not only will you have this full Tyvek suit and the multiple layers of gloves, and the reason for that is because the first layer of gloves, just to touch it, or just to put them on, you have to touch it. And so then you put a second one on that you've put on with the gloves. And then you also have, this is an example of a fume hood, and you have a box in here that you put your arms in. And so that is another way that you can sort of put barriers between you and the sample. Anyways, I don't have too much time to talk about the logistics of this because I want to get into the meat of the talk, but feel free to ask me questions about that later. And then sort of the final step is the computational analysis. And essentially, you get all of these reads, which are represented. The top represents whole genomes. And then you get reads, which are sort of these very short bits of the genome back from sequencing. And then what you do at that point is you align it to a reference genome. So this is whatever you're studying. So, maybe it's a pathogen, maybe it's the human genome, maybe it's cattle or sheep or some organism. You're mapping it to that, and then you can run your analyses. All right, so I guess we'll jump into some examples. So the first one, we'll have sort of a couple of vignettes of a couple different plants and animals that we interact with. And so the first one I want to talk about today is a watermelon. And you can see in this picture all of these really diverse varieties of watermelon today. There's ones that are yellow, there's ones that are red, there are ones that are white. There's ones that are quite big, and there are ones that are quite small, even when they're fully grown. And initially, the wild progenitor of watermelon would have looked more like these small ones. I hope you can see my mouse on the screen. More like these small ones. They're relatively small. The flesh was white, and they weren't as sweet as they are today. They tasted more like something like cucumber. So still maybe delicious, but not as sweet as the ones that we have today. And then sort of over this long process of domestication and selection, they've become these fruits that we more commonly know today. The seeds have become smaller, and you'll even sometimes see seedless watermelons. That's ones where the seeds are so small that you can just eat them. Yeah, so there's been some really cool ancient DNA work related to watermelons. There's a paper here that looks at watermelon from a 3,500-year-old leaf from a pharaonic tomb in New Kingdom, Egypt, which is really cool, by Susanna Renner. And so essentially what they did is I have a figure from that paper, a leaf preserved in this tomb in Egypt. And you can see a picture of it here. And they were able to take a sample of that and extract DNA to look at different properties of this watermelon. And they also sort of note watermelons depicted in tomb paintings in ancient Egypt. So I thought that was something really interesting, that they're physically in the tomb, the fruit itself, and also representations of them as well. And watermelon, just before we get to what they find, was domesticated, it's thought, in North Africa. And this is a map of different varieties of melons and their natural ranges today. But yeah, they found a couple different things about this particular fruit. One is that they found it had the gene responsible for making it red. So it would have been red like watermelon we eat today. And keep in mind, this is over 3,000 years ago. And it also had genes responsible for that sweeter taste. So essentially, it has a mutation that knocks out one of the proteins that makes it a little bit more bitter. So it would have been sweet like today's watermelon. So kind of cool that we could know what people were eating and what it might have tasted like, or get an idea of what it might have tasted like over 3,000 years ago. Another paper on watermelon, which I think is really exciting, shows that 2 different species of watermelon crossed to get us the melon that we have today. That's actually pretty common in domestication, that you'll have multiple species intergressing, that's the term that they're using here, or hybridizing, that's another term you can use to produce some of the fruits that we eat today. And of course, this isn't something that's unique to the plant world. It also can happen in humans. You might have heard that your genome contains some Neanderthal DNA. That's another hybridization or intergression event that essentially these closely related species of humans and modern humans have intergressed or hybridized, and we all carry a little bit of that in our genome. It's maybe 4% or less, so it's not a lot, but it's a little bit. So anyways, it's kind of an interesting phenomenon that pops up pretty frequently. So the next example that I want to move on to is the date palm. The date palm is one of the earliest domesticated fruit trees. It was thought that it was first domesticated over 6,000 years ago in the Persian Gulf region, and later was grown throughout the Middle East and North Africa. And for a long time, it wasn't certain which species of wild date might have contributed genetically to modern day date palms that we eat today. But fortunately, there have been some archaeological remains that have enabled researchers to do DNA tests and get a better idea of this. And so this is also from Egypt. What you see here are 2 jar stoppers. So sort of figures A and B that you see here, and this is a paper by Escobar et al., are jar stoppers made out of fig palmlets or fig leaflets, sorry. And so they were able to extract DNA from these. These are at Saqqara, the Old Kingdom in Egypt. And so these are a bit over 2,000 years old. And so they were able to get an idea of the species involved here. And much like watermelon, it was also a hybridization event that multiple different species of fig or of dates contributed to the ones that were eaten in ancient Egypt and also today. And they also provide us a couple really interesting glimpses into their analysis. So the one you see in figure D is radiocarbon dating that helps us get an idea of when this plant was alive. And then the one in E shows you the damage on the end of a reed. And this is really important actually in ancient DNA. So essentially DNA degrades over time. Over the thousands of years that it's buried in the soil or buried in permafrost or in whatever context it's in, there's sort of taphonomic processes that happen to the DNA. And those processes actually are really important when we're working with ancient DNA. I mean, sometimes they can be a little bit of a nuisance because if you sequence them, it can lead to mistakes. But luckily, there's a treatment you can do to remove that damage. But they also let us authenticate that it actually is ancient. Because one of the first papers published ever on ancient DNA was in 1986. People thought they had extracted ancient DNA from a mummy, and it actually turned out it was modern contamination from the scientists. And now we actually can tell ancient DNA apart from modern DNA because ancient DNA has this damage. And that's what they're showing here, actually. As you can see, these lines are essentially what it's showing is every base pair in the DNA sequence and how much damage there is at that base. And so you can see on the ends, there's more damage. And so that's typical of ancient DNA. So that's good. When you see that, you say, oh, good, it's ancient. It's not, modern, dirt or modern human or modern, some pollen that's blown in. You can actually know that what you're sequencing is ancient, which is pretty exciting. So it's helpful when they put something like that in a paper to show you, basically to authenticate their results. Great, and sort of another exciting thing, also related to dates, this is a different study, is that there were desiccated or sort of dried out seeds from about 2,000 years ago, and scientists were actually able to grow them, to sprout them. So these seeds that had sort of been dormant for 2,000 years from a species or sort of a subspecies of date palm that isn't around anymore, they were able to grow. They were able to grow 6 different plants, and two of them were females. So they're actually able to interbreed them. And study these plants and what they would be like today. And this brings us to a really interesting topic called de-extinction. And so I'll tell you a little bit more about that in a second. But before we get to that, just if you want to learn more about sort of date palms and these ones that were regrown or sort of other ancient DNA projects on them, I definitely recommend checking out the Kew Gardens website. They have great research on this and a great discussion of it. So I definitely recommend checking that out. But it sort of brings us to, because sort of germinating these seeds, I wouldn't quite call de-extinction, but it gets us sort of down that path. And so, you know, there's been talk, I'm sure you've all seen Jurassic Park, about sort of if we have DNA from an extinct organism, should we bring it back? What should we do with that? And under what conditions should we and what conditions shouldn't? So if that's something that is interesting to you, I highly recommend this book. It's called How to Clone a Mammoth. It's written by Beth Shapiro. She studies ancient DNA from the permafrost, so from mammoths and from woolly rhinos and other extinct creatures like that. And this book both tells you how it could be done and also explores the question of whether it should be done, which I think is an equally important question as to whether it could be done. But so anyways, that's just sort of a tangent, but something that you might find interesting and a really good read as well. All right, and so next I'm going to talk about the domestication of wheat. You know, this is one of the largest crops globally today, and it has a story that's even stranger actually than hybridization. So we talked about hybridization, it's different species coming together and intergressing, and this has, something even more, I guess. It has 3 different genomes inside of like a single wheat cell would have 3 different genomes inside. And so I'll tell you the story of how that happened. So wheat was domesticated over 10,000 years ago in the what we call today the Fertile Crescent. I like to think of this as a slow process. That domestication isn't something that sort of happens overnight. It's something that happens over, you know, hundreds, thousands of years, that people are interacting with plants. You know, maybe they're replanting ones that had some sort of beneficial trait. Maybe they're not replanting, or they're pulling up plants that don't have a trait they like. So this might be something to do with taste. This might be something to do with the size of it. This might be something to do with sort of the yield of it, the ease of harvesting, all of these things. But I like to think of this as sort of a long and slow relationship, which I think can explain sort of how these changes can happen, these domestic changes that happen in domestication, that it isn't something sudden, but it's something that happens quite gradually. So just to give you an idea of sort of where geographically wheat was domesticated, this is sort of what is traditionally referred to as the Fertile Crescent. And so you get wheat and other crops being domesticated here. You may have heard of sort of different types of wheat. You may have heard of emmer wheat, einkorn wheat, spelt wheat, bred wheat, and maybe wonder about the difference between some of them. Well, some of it has to do with these different genomes that wheat has acquired over time, but a couple of the big traits are sort of whether it has a brittle rachis, you'll see that written here. And essentially what that is whether the wheat sort of when it's dry, when it's mature, whether it breaks off of the frond, probably using the wrong word now, but whether it sort of breaks off on its own at that point or whether it stays on there. And so wild wheat naturally breaks off. So it breaks off so that it can go, you know, spread its seeds and new wheat can grow. But that's not ideal for harvesting, because that means that if you harvest it, if you wait till it's totally dry or totally ripe, it's already gone, right? It's already scattered. And so it's really great that if, for humans, at least for human interest for harvesting, if it has a non-brittle rachis. So essentially that's where it stays on, and then you can harvest it with a sickle or something. And then you get more wheat. And then sort of the other big change that's happened is you have whole grain versus naked grain. So essentially whole grain is where you have the hull or the protective sort of casing around the grain stays on there, or naked grain, which it's quite easy to get off. So these are some changes that have happened over time. And one of the things responsible for those are multiple species crossing. And I think that's one thing that's really exciting. So essentially what you see here is a little math equation to represent at the end the type of wheat that we consume today. But essentially you had, it's all sort of 3 different species of grasses, but you had 2 sort of wild wheats cross to get these bigger ones that we're more familiar with, and spelt is one of these. And then you have a different species altogether called goat grass. That's what this is, that it crosses with this, and then you get this bigger wheat. And so sort of unlike in the melon case or in the date palm case, where maybe you have some genes, or even in the human and Neanderthal case, right, where you have some genes from this other species, this is where it's taken the whole genome from another species and incorporated it. And so this is sort of a diagram you can see here of, it has 6 pairs of chromosomes of each chromosome. So you've probably heard that humans have 2 pairs of each chromosome, that we're diploid, that we have 23 chromosomes and a pair of each one, so 23 pairs of chromosomes. Well, they have, instead of pairs, they have 6 copies, which is kind of cool. So they're called hexaploid. And there's some fruits that have even more. So for example, strawberries, octoploid, it has 8 of these, 8 copies of each chromosome. Anyway, so I think that's pretty wild. And then I just wanted to show you what it looks like growing. So this is wheat and this is goat grass. And so essentially wheat, stole the whole genome from goat grass, and that's given it some of the characteristics that it has today. All right, so we're almost at the part where I'm going to show a couple of objects. One that I'm going to show is a mortar and pestle, and essentially these are really important for grinding various grains down into a flour that can be used, for instance, to bake bread. And so they're going to be important even pre-domestication when people are harvesting wild ones of these. And so the one I'm going to show you today actually is from the Mesolithic. So it's sort of from this transitionary period into farming, into the Neolithic, into agriculture. Yeah, so maybe I'll stop sharing for a second to do that. Okay, so hopefully you can see just me. I'm going to put some gloves on. And also now is a good time to think of questions. Okay. Great, so this is a pestle, and I hope somebody let me know if you can't see it, okay. But so this is the pestle, and so essentially, as I mentioned before, it's a Mesolithic pestle. So it's from pre-Neolithic times when people were perhaps, in this gradual phase of domesticating wheat, for instance. It's from Iraq, yeah, and it's made of basalt. And you can see, actually, I don't know if you can see this on your screen, but there is some residue, I guess I would call it, on the end. So if a plant geneticist wanted to sample this, I don't know if it's plant, it may just be dirt, but that's something that somebody could study. That's kind of exciting. All right. And so I also have, the opportunity to show you a few other objects that depict wheat. Trying to think of which order. There's a coin I want to show you, but I'll show you the coin at the end because I also have an image of it because it's a little bit difficult to see. This one I think is quite exciting. This is called an Ostracon. And so an Ostracon is ceramic, basically a ceramic sherd that is used for writing. And I'm gonna try and show you the translation or read you the translation of what is on here. So essentially, and I hope you can see this okay, I'm going to hold it from the back so that I'm not touching the writing. But so essentially this is an Ostracon, so it's made of clay that's been baked, and then it's written on in Demotic. So this is from Egypt, and we know it's from Ptolemaic Egypt, and in fact we have a date for this because it contains a date, which is amazing. So it's from the 24th of December, 248 BCE. So yeah, over 2,000 years old. And so essentially what it's saying is it's about the sale of a land, and then it's suggesting that the person then could grow wheat and other things on it. So it says Abucus, son of Seraptos, the agent of the, and this next word is in Demotic, so I can't read it, Lasones, who speaks of, oh, it's a lot of names, of Tothsumia. It is I who said, build for yourself on the parcel of vacant land, the mixed garden, 9, and then the next word is missing, of wheat. So essentially it is a lease of land, and you can see it written on this Ostracon. And it's amazing that we have this over 2,000 years preserved, and we can see this transaction. So I hope you can see it. Okay. Excellent. All right, and then the other things I wanted to show you, we have some rollings that are made from cylinder seals. So these aren't the seals themselves, but this one both shows hunting, and then also one of the figures is holding a shaft of wheat. So you can both see a deer that has perhaps, or some sort of, maybe an antelope, an animal with horns that has been hunted, and then also a shaft of wheat. I hope people can see it okay. I can actually see the screen, so I hope it's focusing. And as I said, this is an impression. So it would have come from a cylinder seal that would have been round and then rolled on clay or plaster. This is plaster, and it's modern, but to show what the seal is ancient, but the cast itself is modern. And then also this is an amulet from Egypt showing a date palm. I know it looks like a pineapple, but trust me, if you look closely at it, this is a date palm. And so essentially amulets could have been worn either by people who are living for various good fortune or for the deceased to provision them in the afterlife. And I actually don't know what context this is from, but yeah, this is an amulet representing a date palm. Great. And then the last one I was going to show you for now is a coin. And then I also have a picture of the coin on a slide so that you can see it more up close. But essentially, this is a coin, a Roman period coin from central Anatolia from a site called Alashar-Huyuk in Turkey. So as I mentioned, a Roman period coin. And so you can see what in theory you can see here is some wheat in the middle. And on the reverse, I'll show you the reverse side as well, is a picture, because remember this is Roman period. This is a picture of Alexander Severus. So you can see his image on one side and then the wheat on the other side. It's made of bronze. Excellent. And so I'm going to reshare my screen so you can see that. May take me a second to reshare. Sorry about that. Okay, almost there. We're not there yet. So you should see my screen and then slideshow. Oh, hang on, sorry. Shared screen's a lot, but okay. Great, okay, so hopefully you can now see the coin. This isn't the same coin that I showed you, but it's the same design. So again, it's Alexander Severus on the front, and then these shafts of wheat on the back. Great, so I hope everyone can see that okay. And I'm going to jump into the next phase of our talk, which is I'm going to talk about lactase persistence. And I find this a really fascinating topic because what we've been talking about before are sort of genetic changes that we as humans have brought about in especially the plants that we interact with, right? For these different traits that are desirable to us. So things like a sweeter watermelon or a bigger watermelon, things like that, or grain that's easier to harvest. But I think it's also really interesting to think about the changes that have happened in us because of domestication. So I'll talk a little bit about lactase persistence. And so lactase is an enzyme that's produced in the small intestines of mammalian infants, including humans like us, and it allows for the digestion of milk, right? So when we're infants, everybody's able to digest milk, but then some people lose that ability after weaning, and that's actually most people. So about 66% of the world's population is lactose intolerant. They can't digest milk into adulthood, and about 1/3 of the world's population is lactose tolerant. So it's not the most common, but it's, a large number of people are lactose tolerant, and it's thought that the ancestral condition is to be intolerant, that sort of like many other species, we lost that ability after weaning. And so essentially it's downregulated and eventually stops. We stop producing this protein that allows us to digest milk. Yeah, and so for a long time, cattle and other animals that we get dairy from, like sheep and goats, were domesticated over 10,000, 8,000 years ago. And so for a long time, it was thought that lactose tolerance was something that occurred around then as well. So at around the same time that people domesticated animals that could be used for dairying. But genetic research has started to call that into question and ask whether other things might have been happening and whether this might be a more recent condition or a more recent trait. And so what's the archaeological evidence for dairying? There's lots of different kinds of evidence. So there's faunal remains. So these are cattle remains that you can see here. There's also herd mortality. That's what you're seeing here, is essentially if you take faunal remains and you look at the ages and the sexes of the animals that are found, that sort of different profiles exist for different, this is sort of in modern day, but then you can apply it to the past. Different profiles are ideal for different uses. So if you're going to milk animals, that might be a different profile than if you're using them for meat, or, you know, in the instance of sheep and goat, if you're using them for wool. So, you know, for sheep and goats, you'll want like a lot of adult animals if you're trying to get wool, and that might be different if you're trying to get meat. Maybe you're, you know, your herd profile is a bit younger, for instance. And then also males and females can be different depending, you know, if you're, for example, if milk is your primary product, you would maybe want more female adult cows, for instance. Yeah, so that's another way that we can get an idea of what people were doing sort of when they were domesticating animals, and then also what was sort of the primary purpose of those animals. There's also residue analysis. So that's what you see here. This is a pot and it has, I actually don't know what's in it. I'm gonna, yeah, I don't know what's in it, but anyways, you can look at that chemically. There are ways to sort of chemically identify, especially when there are residues like this, you can look at the proteins and see sort of what type of food could have been either prepared or consumed in a pot like that. And then there's another new tool. It's kind of similar to ancient DNA, although it's not ancient DNA, it's technically proteomics, looking at ancient protein. And this is a way to see whether people were directly drinking raw milk, because sort of raw milk has a lot more lactose than processed milk. That, you know, things like yogurts, things like cheese, you can actually process a lot of the lactose out of that. And so raw milk has sort of the full amount of lactose. And so there's a really cool thing where you can look at dental calculus. You see this in the slide here. Essentially, this is this like, it's not dirt, but it's plaque that's built up in your life. So, you know, when you go to the dentist, they clean it off. But in ancient times, often it would just build up over the course of someone's life. And you can actually study that to see what they were consuming. You can see if people were consuming raw milk or not. And so in this particular study, they looked at sort of a medieval Norse cemetery, and they saw that they were indeed drinking raw milk. And so it'd be really interesting and exciting, I think, to look at older time periods like the Neolithic, like the Bronze Age, to see if people then were consuming raw milk as well. Yeah, and so what are the genes that sort of provide or sort of lead to this trait? So, you know, a lot of times people will talk about it as if it's one gene or one mutation that allows people to be lactose tolerant, but in fact, it's about 15 different genes or sort of mutations in the same gene that allow this, sort of 15 different variants. And so you can see sort of the 5 most common shown here. Yeah, and they're sort of in different parts of the world. So you can see sort of Eastern North Africa, Eastern Sub-Saharan Africa, the Middle East, and also Europe. So this is sort of evolved separately in all these different places, which is kind of cool, or happen separately. These are these different mutation events. And then because they did let people drink raw milk, they sort of stayed around, right? These mutation events happen all the time. Yeah, but this one is one that yeah, has happened a number of times and contributed to this same trait. There's also images, I really like ones like this that sort of show the different variants sort of in aggregation here. So you can see sort of these blue areas show places where a lot of people have the ability to drink raw milk, and then you can see areas that are lower, and sort of those lower areas, you know, if you're lactose intolerant and you know, yeah, essentially it doesn't mean that you know, there's lots of ways to process out the lactose out of dairy products. So there's still ways to consume it. And I actually think that's something that's really important when we consider the archaeological record, especially because we'll see this is a recent evolutionary or recent mutation. And so actually maybe people in the past were doing a lot of this processing that makes milk consumable, even if people are lactose intolerant. And this, I actually really love this plot. This is from the Mace Lab at University College London, and essentially it shows in aggregate over all these different variants and based on the data source. So they're sort of not making assumptions for whether people are lactose tolerant or intolerant in between, but sort of just showing at each of the places where data has been. So these red colors are places where people, where there's high levels of lactose tolerance, and then these light blue is low levels. And you can actually see some of these places are quite close to each other. So if you look, for instance, at Sardinia and then at Italy, you can see there's a big contrast, even though geographically they're quite close. And as I mentioned before, this is something that scientists think was actually a rather recent phenomenon, that it wasn't 10,000 years ago, wasn't 8,000 years ago, but actually may have been even selected on in the past 2,000 years. This is a paper from Field et al. And essentially this one's modern data. They use modern data from humans in a data bank called the UK Biobank. And they found, they sort of did a scan that looks for a selection of all types. And they found actually this LCT, which is the gene that's related to lactose tolerance, was the one that is most selected in the past 2,000 years, which is kind of cool. They also found the MHC, which has to do with immunity and responses to pathogens. So I thought that was interesting as well. So if that's what we see from modern DNA, what do we see from ancient DNA? And so this is a plot looking at just one of those variants that we saw before, one of the 15 from the Mathiasen lab. It's a lab that does a lot of cool work on selection. And essentially they see in different places, this gene, becomes more common at different time periods. So in green, you can see Britain and Ireland. You can see that it's sort of starting to become more common maybe 3,000 years ago, 2,000 years ago. And then you get to present day levels, which is maybe 75% of people, for example, in Britain and Ireland are lactose tolerant. You can see Central Europe is sort of similar. And then you can see some places that are a little bit dissimilar. So the Indus Valley, for example, in Pakistan and India, or Iberia. So that would be Spain and Portugal. It's sort of, you see this variant, basically appearing later and then increasing in frequency later as well. And so now I mentioned we get to some projects that I've worked on. I had the opportunity during my PhD to work on this project about ancient Rome. And so we looked at the population of Rome through time from about 10,000 years ago all the way through to the medieval period. So sort of spanning the rise and fall of Rome, if you will, to look for a bunch of different things. I mean, essentially we were interested in who was in Rome and was that changing over time. But we also did look at things like selection and lactose tolerance. And so, I really wasn't sure, looking back at this slide, that we see from Mathiasen, would it be more like Central Europe or would it be more like the Indus Valley and like Iberia? And so we said, well, we should, look at this trait in our study samples too. And so this is what we see. So I can essentially show you, this is laid out similar to that Mathiasen slide. So this is time. So this is 0 or 1, I guess we should say CE, so that's 2,000 years ago. This is 1,500 BCE, so that's like 3,500 years ago, all the way back to about 10,000 years ago. And you can see that almost no one, so sort of 0 means, because you have sort of 2 copies of each gene, right? So 0 means that people don't have the allele that allows them to digest milk. And then about 2,000 years ago, you see it start to rise in frequency, but you can see that it actually still, in the present day, is quite low. And that's true across Italy, that about 20% of the population has this particular variant. So we thought that was interesting. So it is sort of more like Iberia or the Indus Valley in that regard. But of course, like I said, there's not just one variant, there's multiple ones. So we decided to look at all of these. And so this is a plot here of those. And you can actually see that 3 of them rise to frequency, or a higher frequency than 0. So essentially 0.5 means that somebody has 1 copy of the gene and 1 means that somebody has 2 copies of the gene. And so what's cool is, Rome was sort of this really cosmopolitan place. That was the other thing we found, is that people were coming from all over the Mediterranean and beyond. So essentially you see, this variant, for instance, appearing in Rome and rising to frequency, as well as this one. So interesting to just see sort of these different variants that have arisen in different places, different contexts, and in the same place, contributing to the same trait. So I thought that was pretty interesting. And there's also been some research about whether people, I mentioned dental calculus before, where you can look at the stuff on people's teeth and see if they've been drinking milk. Essentially, there's been studies on that have told us about what people were consuming in ancient Rome, which is really cool. And so this is a paper where they looked at Roman era individuals in Britain and then Roman era individuals in Italy. And essentially they find that those in Britain are consuming raw milk and those in Rome aren't. So, this may be an interplay between diet and people, being creative and processing dairy in a way into cheese or yogurt such that they can consume it, without, even if they aren't lactose tolerant. And so I'm going to show you a few things related to dairy processing now. And just came back to this slide to show you that it's sort of more like these case studies. All right, so I'm going to show you some strainers that were used to process milk and essentially separate the whey from any curds in cheese making. Okay, so get some gloves again. And so, yeah, what I'll show you, the first thing I'll show you is a strainer. And one thing that I think is interesting about this strainer is that people for sort of a long time didn't know what they were used for. And then I mentioned residue analysis before. This is one of the things that residue analysis has helped us understand what it was used for. And it seems like, well, I'll show you two strainers and they might have been used for different things. I'm careful with this one because it's quite big. Okay. And so you can see here, the strainer. I mean, it honestly looks a lot like a modern colander. I hope you can see it okay. And essentially what this would have been used for is that it would separate whey from any curds that you have when you're making cheese. And so, yeah, I'll show you the base of it as well. And then there's another strainer that I'm excited to show you. This one is much smaller. And so this one probably wasn't used for dairying, but instead would have been used maybe for beer or wine. So after the fermentation process to get either, parts of the grapes or seeds out or sort of any large particulate out. And the one I showed you previously had a base. You can see this okay. So that's the bottom of it. This one doesn't have a base, so perhaps this was sitting or resting on something. Great, so that's most of what I wanted to show you. I guess I have one more artifact to show, but now is a good time for questions. Similar to the Ostracon, this is another wheat example, similar to the Ostracon, there were also a number of clay tablets that recorded transactions, either transfers or payments of things like grain or land that grain would have been grown on. So I can show you, this is a tablet that's written in Akkadian, so sort of a very old and early language. And hopefully you can see this while you're thinking of questions about ancient DNA and plant and animal domestication. There's actually even some writing on the back, just a little bit at the top. There we go, so you can see. Great. All right. If people have questions, I'd be delighted to answer them.
Great. Well, thank you so much for a fascinating talk. It was really wonderful and great visuals to accompany it as well. So we've already received a few questions here. So for anyone who has additional questions and they haven't submitted them yet, please just type them in the chat. So pop up the little chat box and just type them in there and send them to Kirsten Newman and I can read them out to Hannah. So for our first question that we've received, how has ancient genomic data been used to reveal details about the co-evolution of crops and pathogens? Second part to the question, what new information has been gained about the history of agriculture in specific areas from the study of ancient DNA genome-wide sequencing?
Oh, absolutely. I mean, the pathogen question is fascinating because, I'm sure we've all heard of things like the potato blight, or bananas, for instance, have been, sort of historically, especially when grown in a monocrop situation, really susceptible to blight. I'm trying to think of specific examples from the archaeological record, though, of pathogen resistance. That's a really good question. For people I know that we've seen, so this won't quite apply to plants, but for people we have seen selection for variants that are protective against malaria, against tuberculosis, against leprosy, and in one of the papers I showed, the one from Field et al., there was, for instance, the MHC, which is a region that's responsible for immunity, was one of the regions that's more selected on. I imagine certain plants also might have gone through something similar. Yeah, and I think there has even been studies of the potato famine and the pathogens that cause that. So that might be something interesting, even just information about sort of which varieties or strains of a plant are susceptible and are immune can sort of help make decisions about which ones to propagate in the future, which ones to cross with each other in the future. And also just having sort of the record of diversity of genes of a plant that, sometimes when things are grown in monocrop or you're aiming for a very similar outcome or product or fruit, that you can sort of reduce genetic diversity and having a record of sort of the broader range of genes involved might be useful. In fact, there are whole seed banks where they save sort of seeds from diverse lineages.
I've heard about those. It sounds absolutely fascinating. I think it was about lentils and how we've lost so many types of lentils, but these seed banks have them, and sometimes they access them in order to start growing them again. Really interesting work. Okay, another question. First of all, thank you for a very nice talk. The question is, you mentioned the need to wear a lab suit to avoid contamination to the sample, but what about contamination during field work and excavation?
That's a fantastic question. Yeah, there's definitely, in sort of the ideal world, when you're doing field work, and I've done this, essentially that you'll go out into the field, and my collaborators here have done this as well, that you'll wear protective gear. So that might mean a face mask, that might mean gloves, that might even mean a full suit like that. And that when excavation is happening and then something that might, you might want to test for ancient DNA is uncovered, that then you can, essentially take an aliquot or a sample of it right then and there. A lot of times that's not the case. So for instance, on my PhD, a lot of the material I worked with was housed in museum collections. So it had been handled often and over a long period of time. And we found that those didn't have a lot of contamination. I think one thing that really can contaminate it, and sometimes happens at field sites, is washing something in water. That if you dunk something in water, that's sort of not ideal, because then all of a sudden sort of any red DNA that's in that water is getting pushed into the material. So yeah, as long as I said that once to somebody who works on shipwrecks, and he was quite sad, which made me feel bad. I was like, just don't get it wet. And he was like, oh. So anyways, yeah, so I mean, that would sort of be one of the bigger things. And then also consolidants. So if you have glue on it, that's another place that you can introduce, a glue, especially if it's made from animal sources, then we'll have DNA from that animal. So, yeah.
So many avenues to approach it. For the strainers specifically, do you know, or would they have been lined with coarse cloth? The holes in the strainer seem rather large, so a lot of things understandably would have passed through them without that type of material. Do we have any evidence of that?
Not that I'm aware of, but I imagine, that's a really good suggestion. Yeah, and especially for that small one, the holes were, quite, they were almost as big, well, not almost as big, but they were still quite large, and so, yeah, I imagine something like that. I don't know of any archaeological evidence for that, but that might be a case where sort of modern practices today could inform that. Yeah.
Mm-hmm. Absolutely. Is there any genetic evidence for when and where woolly sheep were first bred and how they spread around the world? This might not be your area of expertise, but I'm throwing them at you anyway.
Oh, I don't know. Yeah, I actually have no idea, but you should look that up. Sheep were domesticated in the Near East as well, but I don't know about sort of, and there were spindle whorls. Yeah, people were using sheep for wool, in the Near East, but yeah, that's a great question, because there also are wild sheep sort of in a bunch of places, and they may, like other species, have had multiple centers of domestication. That if you look at cattle today, it seems, that cattle from sort of the Near East and North Africa and Southeast Asia are all contributing to cattle diversity today. So I don't know, that's a good question, and I hope someone's researched that, because I think it's really cool.
So kind of related to a question we often get regarding the museum collection and antiquity, what is the oldest ancient DNA that has ever been sequenced?
Yeah, that's an awesome question. So there's a paper, I think it was in the past year actually, the Beth Shapiro's labs, that's the person who wrote that book that I mentioned, the How to Clone a Mammoth, her lab sequenced I think, it was Mammoth DNA that was over 1 million years old, which is just mind-boggling. Like, yeah, so that's amazing. So, and that is sort of ideal conditions, right? It was preserved in permafrost, and essentially that means it's frozen and that it's not thawing, because that's one thing that can really destroy DNA is if it's going through freeze-thaw cycles. So permafrost is sort of the ideal conditions because it's not freezing and thawing and freezing and thawing. It's just frozen. Yeah, but I think that was much older than people maybe thought was possible. I think before that it had been maybe 400,000 years. So that was more than doubling. Yeah.
That was still very old from our perspective.
Yeah.
Okay, we have a question from an attendant who is doing her master's degree in geoanthropology.
Oh, awesome.
I can't even say that. Geoanthropology at Istanbul Technical University.
Yeah.
So her question is, how much do excavation bureaucracies affect your work or the work? For example, when you collect a sample from such places as Iraq or Turkey, can you work on it right away? Does it take a long time? Are there restrictions on it?
Yeah, that's a really good question. It really depends on the place, but I think it is sort of best practice to make sure that you're working with any local authorities, right, on permitting. So sort of the place that I'm most familiar working in Italy, it was a team of people. So our sort of research team was people from Italy, and then also from Vienna and Dublin, where the ancient DNA was, and then also from our university here in the US. But so essentially, for that, we worked with the Italian government and the Italian superintendents to get permission for each of the material that we worked with. And we also worked with archaeologists from each of the sites. So I would say, I guess the bureaucracy differs, but it's something that is good to start out with. And I think is, even sort of, if possible, before excavations, so that sort of, permits are in place when you've already started working, so that so that you could go to the field with collection material if possible. That's a really good question. Also, feel free to message me. I'd be happy to talk specifics as well. Yeah.
So another question, do you have any thoughts or comments on practices with ancient DNA and human remains?
Yeah, absolutely. That's a great question, right? Because there's sort of extra, you want to take extra thought into preparing to work with human material. And so I think you'd certainly want to identify any stakeholder communities and engage with them about sort of, is this a desirable thing to happen, right? This ancient DNA research. And the answer to that might be no, and then that's fine. And then you don't do the research, or the answer might be yes, or there might be some sort of in between that, yes, but there are conditions. And so sort of research in different parts of the world has happened in those different ways. One condition might sort of be how that DNA gets used. For instance, sort of, is that for that specific study, or is it publicly available for anybody to use for other studies? There was a case, sort of a famous case here. It's with modern DNA, but I think it's very relevant to ancient DNA as well, where the Havasupai tribe in Arizona gave DNA samples for a study about diabetes, but essentially then so people gave blood samples, and they consented to blood samples, but they didn't consent to other sort of for diabetes research, but they didn't consent to other studies. And then that material was used in a whole bunch of different studies in ways that was harmful to the community. And so that would be a case where both, you'd want to make sure that you're engaging with stakeholders beforehand, and also considering sort of the implications of the work that you're doing. So I mean, that's a really good case to read up on if you're sort of interested in sort of the implications of working with either modern or ancient DNA. And I think I think it's good to remember that, even if something is ancient, that it can still really impact people in the present. And so to treat it as such, where you have IRB processes and other processes that sort of make sure they're protecting the people who are, whose DNA this is.
Yeah, those are really, really good points to keep in mind. We have one comment that Dan Bradley, TCD Dublin, has a new project on the rise of Bronze Age wool using ancient DNA.
Oh, excellent. Oh, that's exciting. Yeah, that lab does some really cool work. So that's awesome to hear.
One other question, do you know of any examples of extinct edible plants?
Oh, I'm sure. I don't know if I know of any off my mind. I mean, even that date that the date palm that we talked about earlier, I mean, the one that was regrown that species wasn't around anymore, and then was regrown. But I'm sure there are other edible plants that unfortunately haven't made it today, especially sort of with fewer and fewer species sort of being the ones that are cultivated. One that is edible, but we don't eat in the US so much is something millet. So I studied millet. So don't get me off on millet because I'll talk too much about it. But, millet people eat, in lots of parts of the world, but not in the US so much. And I think, it's sort of sometimes it's these weird historical reasons. But so I think there's a lot of crops like that, maybe are a food crop somewhere and not elsewhere, and then some that have fallen out of use. I'm sure I'm going to think of a good example after this, but yeah.
From your hopes or kind of your experience in the field, where do you see the studies of ancient DNA going?
Oh, that's a really good question. I think several different directions. I mean, one thing that I'm excited about is, for example, studying dental calculus or studying soil. So these sort of non-destructive directions where you're getting, this interesting information either about plants or animals or humans. But from these other substances. There was even a study recently, sort of chewing gum. I think it was, 7,000 or 8,000 year old chewing. It was sort of a tar that somebody had chewed, and then they were able to extract human DNA and then also pathogens from that, which I thought was, that's sort of a niche, that's not, a wide field, but also beeswax. So I'd say that's really exciting. Also, I think the coevolution of somebody asked earlier about the coevolution of plants and pathogens. I think humans and pathogens, that'd be a really interesting topic to study too. So.
Great. And one last question. Are there certain environments that are better for ancient DNA than others?
Yeah, that's a great question. We talked about permafrost earlier. So permafrost is really good, and that's sort of you're keeping, whatever sample frozen for a long period of time. And there were certain conditions that people sort of thought it'd never be possible to get ancient DNA from. I mean, even places like Italy, because it gets quite hot there, but then gets cold. And so sort of those temperature swings aren't great. So it's definitely not as good as somewhere as frozen all the time, but surprisingly, even places that are desiccated, quite dry, even if they're warm, can be, good for ancient DNA preservation. But stable and cold is the best, and sort of hot is the worst.
Well,
awesome.
Thank you very much. This is absolutely fascinating, and thank you for fielding so many questions, and to everyone who attended for submitting them. I think we continue to learn a lot more, and I did put in the chat a link to register for our next virtual talk from the museum, which will be on April 26th, connected with our special exhibition. So you can register for that on Eventbrite. And we encourage you to follow the OI Museum and to sign up for future talks and programs. And thank you again so much to Hannah for sharing her research with us this evening. And with that, we'll say good night.
Thanks for the great questions.
For over 100 years, the OI has been a leading research center for the study of ancient Middle Eastern civilizations. Join us in uncovering the past and learn about the beginnings of our lives as humans together. Become a member by visiting oi.uchicago.edu/member.